Well, hello everyone, and welcome to 17 Minutes of Science. We are back again. The weeks are flying by. I am so delighted to have a guest today, Dr. Yamina Bershish, joining us. My name is Sarah Cheeseman, and I am a technical solutions scientist at In Vivo Biosystems, and we have just had a wonderful run talking to people all around the world and right in our backyard about what they're doing in their fields of science. So Yamina is with us today, and I am starting my timer. Look, I was about to run away with myself, but I just started my timer. There goes our 17 minutes. Yamina is the founder of Dr. Gpcr, which is a fabulous name, a platform to spread information on targeting GPCRs, as well as opportunities for various stakeholders from academia, pharma, biotechnology, to form meaningful partnerships and ultimately improve human health. And she is here with us today to discuss more about Dr. Gpcr and what it has been like getting this collaborative up and running. And now she's reaping the rewards of impact. So before I turn it over to Yamina, I wanted to say I was looking at her website, and she has some words to live by on her website, which I really love. So the goal is not to be perfect by the end. The goal is to be better today, by Simon Sinek, who always has really interesting things to say. So on that note, not perfect, just better. So I'd love to introduce Yamina and ask her to say a few more words about herself, and then we'll get into the questions.
Hi, Sarah. Hi, everyone. Thank you so much for having me with you today. I'm very excited for these 17 minutes. Thank God you started the timer. Otherwise, we would be going on and on and on, although it can be a good thing as well. So I am a biochemist by training. I first started working on GPCRs as a summer intern after a class as an undergrad at the University of Montreal, just to make sure that everybody knows what a GPCR is. So GPCR stands for G protein coupled receptors, and they're a family of genes that control almost everything and anything in the body. So you can see, smell, and taste thanks to GPCRs. And for the past almost 20 years, I've been fascinated by these proteins. And I've studied GPCRs throughout all my my graduate studies, as well as my postdoc. And I'm very excited to be here today.
That's great. And thank you for defining it. I was going to ask you to do that because as we were discussing offline, PCR is on everyone's lips, but this is not the same thing at all. So we want to make sure we are all on the same page. So let's jump into the questions and tell us about the founding of the Dr. Gpcr organization, which is quite new as I understand it, and feels like, from what you just said, kind of a culmination of your life's work.
Yes, so thank you for that question. So the idea came sometime in February, and it started out with just the idea of creating a podcast and talking to my goal right now with the podcast was to talk to everyone in the field and interview them so that we can learn more about the work that they're doing and try to spread their research across the globe and use a new avenue to do so. And then the podcast idea evolved into an ecosystem idea where I said to myself, why not have a platform where everyone who is a stakeholder in the GPCR field can come on and we can meet each other, we can collaborate with each other, and then we can together advance GPCR drug discovery. And the interesting thing about it is that GPCRs are involved in many different diseases. So we're not focusing on a specific disease area, but we're focusing on a family of genes that are important in so many diseases.
So you saw a need and you ran right into it and decided to create something.
It's, I'm a scientist by training, so it started out as an experiment, and I said to myself, let's just ask the community. I trust in the wisdom of the crowd, and there has been a tremendous support, a tremendous interest, which, as you know, it motivates me to get up every morning and do the work that I do.
That's fantastic. So speaking about these proteins, the GPCRs are a large family, as you mentioned. It's like they do all sorts of important jobs. So they're a large family of protein targets that are for approved drugs. And so why don't we hear more about this family of proteins, since it's seemingly quite ubiquitous in terms of their functionality and targetability?
So I think we do hear a lot about them, but you have to listen carefully and to go where people talk about GPCRs. So there is about 800 GPCRs. 400 of them are olfactory receptors, and 400 of them are GPCRs that fulfill multiple functions. Out of those 400, there's currently about 150 that are either already targeted in the clinic with drugs or are under investigation. But there are about 300 of them that still need to be understood, functionally speaking, but also could be used as drug targets. So just to give you an idea, when I started out Dr. GPCR, my first question was, where are the GPCR folks? Where can we talk to people who work on GPCRs? And it turns out people are on Twitter. There is a very active GPCR community on Twitter. You can come on Twitter, type in hashtag GPCR and find everyone who works on the receptors over there. There is a lot of chit chat, a lot of helpful information coming and going there. And you can also follow us on GPCR on Twitter at Dr. GPCR.
Well, that's a great plug. Lots of things happening on Twitter. It's nice to hear about the good things happening on Twitter.
Yes, and these are definitely great things happening on Twitter.
How many people or groups are part of the community now that you're building?
So far, when we look at the number of followers on Twitter, which most of them, I want to say 95% of them are linked to studying GPCRs, we're about 800. I'm pretty sure that there is more than that. We're also working on creating a master, yellow pages type of thing where you would catalog everyone working on GPCRs, because if you want to collaborate and you want to get to target GPCRs in a drug discovery context, but also understand their biology, you need to know who to talk to if you have questions.
It strikes me also that in this very particular moment and year that we're in, where we're not necessarily convening to have scientific meetings and discussion, that this has an added importance that you're creating this at this time.
It does. And believe it or not, I am happy that although we are all at home, or most of us who can be at home and work from home from a computer are still active, there is a high number of publications on GPCRs that come out every month. We do have a monthly newsletter. You can subscribe to it at drgpcr.com/newsletter. And actually our August newsletter came out this morning, and we cataloged over 60 research-focused papers on GPCRs on various topics. So you had orphan receptors, you had structural insights on how the receptors function, you name it, you find it there. Wow. The other cool thing is that there are a lot of new and budding virtual meetings that are coming up, including ours. And I'm going to use this opportunity to plug it in. So we're also organizing a free virtual summit, drgpcr.com/summit-2020. Where so far we've had over 300 people sign up to come and talk GPCRs.
That's awesome. And when is it for the?
That's a great question. Actually, it's going to be the week of September 14th. So it's a week-long event, September 14th to 18th, and it's a new type of event. It's kind of the way we think about it is kind of a Netflix a week, where no matter where you are in the world, you can just hop in and watch the talks, leave comments to speakers. And the beauty of it is that it's free.
That is a beautiful thing. And that it's inclusive. So people can come from everywhere.
It is. And can you believe it? The time difference between Boston, where I am right now, and Australia, not Australia, but New Zealand is 16 hours. So they're already tomorrow.
Right, I know. So you can enjoy time warp conversations.
Yes.
Virtual meeting.
Yep.
That's great. Thank you for plugging it. And we will make sure we follow up after this with the information so people can grab that if they want to join you. A question came in from an audience member, which I think is just a variation of something that was on my list. So I'm going to go ahead and frame it in the way that this came in. So as you've described, GPCRs are among the largest family of proteins. There's a huge diversity of receptors. And so how, in your opinion, can they help speed up GPCR discovery, given, I guess, this huge diversity? Like, how does that all work together?
So it's interesting. It's an interesting family of proteins. And although they are diverse in they are categorized in different families. They have different functions. They have patterns of expression throughout the human body that can vary depending on the receptor, but they do have commonalities. For example, the 7 transmembrane domains. So they actually are most of the time expressed at the cell surface, and they have 7 transmembrane domains, and which allows the receptor to be functionally, to be able to detect the signals from outside the cell and then relay the information inside. But that requires a conformational change, so a movement in the protein. So a lot of scientists dedicate a lot of time on understanding how these signals are captured from the outside of the cell and transmitted inside the cell. And we've done tremendous, we went to the future now, so because now we developed tools in the lab to better understand the function of these receptors. We have better animal models, system models to understand the function of these receptors, but we also have bioinformatic tools, which allow us to better and faster analyze the amount of data that we can now today acquire. What I'm thinking about is well, when I started working in the lab, we could only measure the effect of a handful of drugs because you could do the experiment with 24 tubes, for example. And now you can run plates and you can just stack them up and then leave. And you can do these experiments in 15, 35 plates. Mm-hmm. And then you acquire much more data, which means that you can very nicely put these together. But thanks to the bioinformatic tools, we're making great progress.
Something else you just said sparked a thought. So you mentioned about model systems. I mean, yes. Our company is very interested in using models to accelerate discovery, whether it be drug discovery or basic biology. But I imagine you have representation from all different types of models in this field.
Yes. Definitely mouse models, but also some zebrafish models that have been used to study GPCRs. And I think being able to study the receptors at a molecular level is very important, but then in a way putting them back in an entire system, a live animal, for example, where you can look at the receptor not on its own, but as a global part of that system is also very important to understand its role in humans long term.
That's definitely how we approach the world. So like-minded in that regard. Whole animal models are fabulous when combined with other tools.
And we haven't even talked about this before.
Yeah, right. How well conserved are the proteins across animal phyla?
It depends on the proteins. The example that comes to mind, if I remember correctly, I think it was the olfactory receptors. And there's about 1,000 olfactory receptors in the mouse, and there's 400 in the human. So it really depends on where you're looking at. There's other receptors called chemokine receptors, one in particular. There's 3 variants of one in the human, and there's just one in the mouse. So it really is dependent on which receptor you're looking at.
Okay, fair. Well, we can't talk to anybody on this show without asking about how COVID is affecting your world. So let's talk about that for just a quick second, although you might have alluded to this already, but it's affecting us all. So how is your work and the work of those in your network being effective? Sounds like people are publishing a lot of papers.
Yeah. A lot of people are doing what one of my podcast guests, Pauline Sell, said, armchair science. But others are taking the time to reflect, taking the time to compile and analyze data, to put manuscripts together, but also to think about the future. And in us, but also including all the community, the GPCR community, and especially those I spoke to, are very focused on the safety and on the health of everyone in the team. And the goal is to, practice physical distancing, but not social distancing, be able to communicate, to connect and support each other. And I think that very much reflects what's going on in the GPCR community.
I couldn't agree more about the social distancing term. I think we might have used the wrong words when we were first describing what we really meant by that.
I believe so.
Yeah. Well, it's because we're richer for being social in this way.
Yes.
So I wanted to shift gears in our last couple minutes about the fact that you have earned a scientist MBA certificate from Cheeky Scientist, which for those of our viewers don't know what that is, I'd love if you could touch on what that is and just tell us about how you ended up on that journey from academia more to an industrial orientation.
Yeah, so very happy to do that. I've been an academic where I was an academic for a very long time. And then one day I opened my eyes and said to myself, what else is out there? So I joined this association called the Cheeky Scientist Association, and the goal is to help PhDs transition out of academia into industry. And one of their programs was the Scientist MBA. And I was very interested into the business aspect of science. And so this certificate allowed me to understand the basics of the business side of science. It is designed for PhDs. So you learn basic concepts and you can have conversations with non-scientists about, for example, mergers and acquisitions, or the difference between finance and accounting. So really basic concepts, but still are a very good foundation to move ahead and a little bit switch gears, not leave necessarily the scientific part of everyday work, but also open your horizons to new things.
And there's a whole world out there.
There is. You can do a lot of things in life, and with a PhD, the sky's the limit.
Yes, and I think that's such a wonderful message to drive home for people who might be listening today or sharing this with their friends. That is not a message that we just can't say it enough, that there's lots of different things to do. And I love the notion of an organization called Cheeky Scientist. How long did it take for you to acquire that certificate for people who might be interested?
I took my time. So it's composed of about 8 modules. At the end of each module, there was an exam. And at the end of the entire program, there's also an exam. It's a self-paced exam. Some people go through it in a couple of weeks. Others do it in a couple of months. I took my time doing so. I actually went through the program twice because after a couple of months, I totally forgotten what I've studied in the beginning. But still, it was a great adventure, and it did open doors.
And now here you are, I'm sure, applying those skills in your newly found organization.
Yes, definitely.
Well, it's come full circle. Okay, in our last 10 seconds, I know you like to make soap for fun.
Yes.
So what can you tell us about that? You have time to do that right now, or you're just so busy?
So making soap is fun. You can make liquid soap or solid cold processed soap. I know we're almost done.
It's okay. I want to hear this.
It's basic chemistry, and you go on Google, you find what you want to do, and just let me plug it in. I did create a line of 6 soaps that are completely unique, new recipes. So it's kind of like baking, but you shouldn't be eating the soap, the end result. But I haven't made soap in a while because at some point I made so much soap that it's going to be enough for the next 5 years.
Oh, and you're giving it away to friends?
Yes, exactly. So no one is getting any other gifts than a box of soap from me.
Well, right now, that is entirely appropriate. So
absolutely.
Your hobby really jives with the cultural norm of the moment.
Absolutely. Absolutely.
Yamina, thank you so much. This has been a treat to hear about all the things you're involved with. I encourage our viewers to check out her website, check out her organization, join her meeting, and we'll also put all that information up on our Facebook page as we follow up on this event. So thank you again, and all the best with everything you're doing.
Thank you so much, Sarah. So thank you for having me. I really enjoyed our discussion.
Great. All right, everyone, we'll see you next week. Take good care.